So if you look at my cursor, you can see this is the Center for Applied Neurogenetics at the University of British Columbia. This is where Emil and I work. We work as part of a team. We're really an interface between genetic discovery and translational or functional neuroscience. And we are housed up in this top corner. And yeah, let's get going, I guess. So really the message we're trying to convey today for everyone that's attended is to provide a very brief history of the genetics of Parkinson's disease. That is, genetics as a diagnostic and prognostic tool, and how this may be used to help identify particular patients for particular clinical trials, with the end game being therapeutic intervention. From there, we're going to move on to illustrate how genetic information can be used to, model disease from a basic point of view, and how we use particular models. In this case, we use mice, and how they may recapitulate stages of disease progression. And lastly, we'll move on to talk briefly about work that we're doing focused on reversing or trying to implement particular drugs from a neuroprotection point of view in a particular mouse model that we've generated. All right, so we'll start.
Yeah, so hi everyone. So my name is Emil, and as Jordan said, we're postdoctoral fellows here with Matt Ferrer at the Center for Applied Neurogenetics. So I'm going to focus on the genetics of Parkinson's disease. I'm going to give a brief background to it, and also how we can use genetics to infer, to create model systems, but also infer that to non-genetic causes of the disease. So I mainly work in this area here, where I work on population and family genetics. So I'm trying to identify causes of the disease. I try to use that information to inform neurologists, clinicians, how that can actually be used to stratify patients, how we can use it as a diagnostic tool, but also as a prognostic tool. Because as most of you are aware of, Parkinson's disease doesn't look the same from one person to another, but there are ways for us to kind of predict how that's going to progress, and that's kind of where I'm working. And then I'm going to kind of leave that to Jordan then to explain how we use this genetic information to create a model system to kind of then understand how the disease starts, how it progresses, what's happening, and with the main goal of them finding drugs to treat or even halt the disease. And so that's basically it. So to give a brief background, so when I talk about genetic discoveries, there's traditionally two ways you can do that. One is by association studies. So when I say that something is associated with a disease, what that really means is that a certain variant of a gene, as you can see here, this is depicted as C and T. So this is a single nucleotide in a gene, so a single point in your genome that is more prevalent in this case, a disease more prevalent in cases versus controls. It's important to keep in mind that association is not causation, but what it does is that it says that this thing here is more prevalent in this group of people that has a certain disease, in this case Parkinson's disease, compared to controls. I'm going to go back to this a little bit later on. The second approach is familial studies, so that's mainly my work, and that can be done by, traditionally is done by two ways. One is called a linkage study. So a linkage study is that you interrogate how something, a certain trait transmits within a family, but you can also then look at genetic information, how that's been transmitted within the family, and if that then correlates with the disease that you're looking at. And that can be done either by, as I said, linkage analysis or exome sequencing. So the exome consists of all the exons in your genome. So the genome, only about 1 to 2% of your genome actually encodes for proteins, actually has, as far as we know, a function. And exome sequencing then is to sequence those parts of your genome that actually translates into a protein, and then finding certain variants of that protein that then causes disease. So So what this means is that when you do an association compared to familiar studies, that an association is going to nominate, as you can see down here in the bottom, common variants, but they have a relatively small effect of the disease. And what I mean with that is that the contribution to disease might just be minimal, although important, but might be minimal. One exception obviously is, for example, ApoE, which is a main contributor to Alzheimer's disease, which is a relatively common variant. But I'm also depicting here is that on the left side is what I call penetrance. So what I mean with penetrance is that the proportion of people that have a particular genetic change, so it could be then as I said, a nucleotide that's been substituted, and how the proportion of people that has that substitution that also gets disease. Because just because you have a mutation does not mean that you get the disease. There's no 100% correlation here. However, when you look at rare variants, which would be the ones up in the top left corner here, they are very, very rare, so we don't find them very often, obviously. They are very helpful in diagnosis of disease because they all have a higher penetrance, meaning that they are more likely to result in a disease state. Most commonly, these type of variants you find in rare diseases. And Parkinson is not really a rare disease, but what we know is that Parkinson, there are monogenic forms, meaning that there are rare causes of Parkinson's disease. There are rare genetic mutations that causes Parkinson's disease. But these might look a little bit different from what we call idiopathic disease. What I mean with that is without a known cause. And just briefly to explain what allele frequency is, because I kind of keep repeating that. It is how common a certain allele or a variant of a gene is in the normal population. So Parkinson's disease has long been seen as a non-genetic disorder. Everyone thought that this is environment, there is no genetic cause behind the disease. And it took till 96 where the first mapping of a gene, where they did a linkage analysis in a large Italian study. I know you can't really see the family in the middle here, but it's a large family from southern Italy. They found a region on chromosome 4 that transmitted with disease in this family, and that's what I'm depicting on your right there. However, it took about 1 year of a lot of work and sequencing for them to actually find that substitution, that mutation within this region here that actually caused disease. So this was a mutation in synuclein. About 6 years after that, we found that multiplications of the gene, meaning that you have multiple copies of the same gene in your genome, so this has been duplicated through meiosis, also caused Parkinson's disease. And this is work by Matt Farab, and he was a postdoc in Florida. What is interesting about synuclein is that, as you are all aware of, what we see in the brain of patients with Parkinson's disease, you see a loss of pigmented neurons of the substantia nigra. So these are the neurons that produce dopamine. And you can see the dark here is in a healthy brain, and the one to your right is in a patient with Parkinson's disease. But in the surviving neurons in this substantia nigra, we see accumulation of proteins. It looks like big aggregates, and those consist of the protein synuclein, meaning that the gene that was identified, the product, the protein that translates into, is the protein that accumulates in the brain of patients with Parkinson's disease. So this was a major discovery. So after that study in 2003, they went back to a family. So this is a large family. There's one branch is living in southern Sweden, and the other branch is in the US, in and around Arizona. So this was actually described in 1949 by a clinician called Henry Mjönes, and he talked about his family that had aggregated Parkinson's disease, which was very uncommon at the time. People did not believe that it was a heritable disease, right? And since then, we found 59 families with multiplications of synuclein, and this all over the world, including Japan, Korea, to US. We found them in Canada and in Africa. So they exist everywhere. But this is not the only genetic cause of Parkinson's disease. So what I'm showing here is on the top, so it says dominant. That means a dominant disease trait, meaning that you have a single mutation that results in disease or increased risk of disease. And synuclein was always the first one in 1997, but there's been multiple others after that. And the most common one here, LRRK2, is actually explaining about 30 to 40% of all Parkinson's disease patients in Tunisia and Middle East and in Israel. So it's a major contributor to Parkinson's disease in certain areas. In Canada, it's about 1 to 2% of all patients with Parkinson's disease that has a mutation in LRRK2. So LRRK2 is an abbreviation. It means leucine-rich repeat kinase 2. It's a kinase active protein. And on the bottom here, you see something called recessive. So when I say recessive, that means that you need to have 2 copies of this mutation or variant, meaning that it has to come from 2 sides of the family, one from mom and one from your dad. These are relatively uncommon, and they normally result in a very earlier onset disease. You get it around your 30s. And at the very bottom here is what I call atypical Parkinsonism. So these ones result in Parkinsonism, so they result in tremors, bradykinesia, postural instability, and so on, but also additional symptoms such as could be psychiatric problems, it could be cognitive problems, and so on. But we also have, as I explained before, the association studies. So we have about 92 variants that is associated with Parkinson's disease. And as I showed before, these can then be put on an axis. We have the monogenic forms on the top, here in your top left, the ones that have a high penetrance and are rare, very rare causes. And then you have the more common ones from an association study like this down here at the bottom. So that's contributing less to the disease. So as I said, there's about 92 of these loci that's been nominated now. But what's important to keep in mind, and I keep repeating this, is that the individual contribution is very minimal. So what's marked in red here is what's called odds ratio. That means that if you have this substitution, this variant, how much does that increase your risk of getting disease? And 1 being no change from a control cohort, a positive value increases by that percent. So the second one says 1.08. That means that you have an 8% increased risk of getting Parkinson's disease. But then again, only about 1% of the population will have Parkinson's disease, even less than that. So this means that 1.8% is your risk. So it's a minimal contribution. And what's in blue here is the actual frequency of the allele. So 0.0315 means that 31.5% of people has this alternative gene. But what's more interesting about this, so maybe the individual contribution of each of these variants isn't that great, but you can still use them as a cumulative score, meaning that you can look, and here's an example of synuclein again. This is a study that we published a couple of years ago now, 2 years ago, I think. But we looked at Parkinson's patients or Parkinsonist patients that have either no cognitive impairment, mild cognitive impairment, or actually cognitively impaired. We did an association analysis only on the synuclein loci because this is the main contributor to Parkinson's disease from all of association studies. And what we could find here is that on the left side here, on what we call the 3' end of the gene, we find a big region that associates with Parkinson's disease with no cognitive impairments. These are the ones that had disease for a long time that does not develop any cognitive dysfunction. On the other side, what you can see, a small region in the middle here, it's actually 11,000 nucleotides. It's a region that then is associated with cognitive impairment. So these people that has this alternative form of the gene, they do readily develop cognitive impairment. Most of them actually has dementia with Lewy bodies. And this is kind of a interesting way where you can actually use all this association data to kind of predict the progression or predict the symptom onset in patients on an individual basis, but probably works better on when you look at it as a population. And as mentioned before, Parkinson's disease doesn't really start when you start feeling your left hand tremor, for example, or you could be dystonia in foot. It's believed to start about 20 years before that, maybe even longer. It could start with things like constipation. You can have sleep problems. It could be depression. And what we know now is that during this time, as you can see on the left there, we start seeing changes in the brain. What I mean with that is the aggregated protein we see is believed to start accumulating, and it starts in your motor nucleus, so it's kind of down almost in your spinal cord, and this is believed then to progress vertically up to the midbrain and then cortex. But you don't really see this full spread disease until very late on in the disease, and you don't see it in everyone. There is no, as I said, there is no perfect correlation with this. But what we do know is that all the complications, all the symptoms, there's a lot of individual variability and the response to drugs and so on. And to further complicate this, even early in the disease, there's a lot of different causes of this movement disorder. So Parkinson's disease is the most common one, but with this at least 20, if not more than that, other causes that can look like Parkinson's disease early on and then develop into something else. And this is, here again, we can use genetic information, and this is a study we did about 2 years ago as well, where we then can see correlation with different genetic mutations and the symptoms it will result in. And this is kind of giving us a roadmap, knowing that, okay, this patient has a mutation in here, and we expect them to the disease to kind of progress in this fashion. And you can then plot that, as I've done here, where you actually look at how different are they to what we call the typical Parkinson's disease. So that's what you see in the middle here. You see synuclein mutations here. They are generally a little bit earlier onset of disease, and there's some other complications that might not be seen typically in what we call non-genetic forms of Parkinson's disease. But there's also other very rare causes that are very different. Although at the very first instance, when you come to a clinician, it might be really hard for them to distinguish these symptoms and these diseases and the genetic cause. But if you then know early on in the disease, there's things you can do. You can start by what I've kind of like simplified here. You can look at the onset. So having a mutation in any of these genes is going to give you an earlier onset of disease, and when I say earlier, it's normally before age 45. And on the right, you can see then certain genetic mutations that causes you to have a later onset of disease. Do you have supranuclear palsy, or do you develop cognitive impairment? If not, and all of these things can then be predicted to some certainty with knowing the genetic buildup of the disease. And this can then inform the clinician in terms of what care is needed. Would this person be a good patient to include in a clinical trial? Because one major problem in clinical trials is that if you have a very heterogeneous disease, which Parkinson's disease is, you're going to have a wide response to whatever treatment you give in, and it might be really hard to actually tease out if it's working or not. And therefore, it's favorable, not in terms of just money, but also in terms of how many people you need to enroll, to have a more of a homogeneous form of the disease. Hence, we normally include patients with a certain genetic cause because they tend to look more similar to each other than if they're to other genetic causes. And what I'm trying to show here is also, as I said, having a mutation in any of these genes is gonna result in an early disease. And most of these are all recessive forms, meaning that you are two mutations in the same gene. Then the mean age of onset would be around 30, 31. These forms of the disease are highly penetrant, as I showed, so having a homozygous or mutation in both alleles will result in disease most commonly. However, this disease is generally milder, it's very slowly progressing, it responds very well to levodopa. So it's kind of a tighter genotype-phenotype correlation, if you will. What I mean is that knowing the genetic cause and how the phenotype or the disease will look like correlates much higher in this type of disease. However, if you look at the late onset disease, so that here we have two of the mutations that Jordan's going to talk about, DNAJC13 up here, which is, I think he will be explaining about this protein later on, and the VPS35. They result in a later onset disease. Mean age of onset is around 60 for these, but they're also not 100% penetrant, meaning that having the mutation does not necessarily mean that you will get disease. I think, as you can see here, at 70 years, for having mutations in DNA GC13, it's only about 50% chance. On top of that, having different mutations within the same gene is also going to look different, meaning here is an example of LRRK2, as I mentioned, is the most common cause of genetic Parkinson's disease. There's a wide spread of age of onset and penetrance for those as well. And also depending on the population you're in. So this on the right here is, again, LRRK2 mutations in Tunisia and Ashkenazi Jewish populations here, mainly from New York, which has a very similar age of onset and penetrance. However, this yellow curve, as you see to your right, is patients from Norway, and these have about a 12, almost 15 years later age of onset with the same genetic cause. So there is obviously a lot of variability. But to go back to this familial forms of disease. So this here was the very first sequencing study where we actually found a genetic cause of Parkinson's disease. What I mean is that we did exome sequencing in multiple families of late onset disease and identified this VPS35 mutation. So this was done independently by two groups. So one group is us, which we studied a big family from Switzerland. You can see on top. And the second group is a group from Austria, which studied Austrian families. And we found the very same substitution. And just to put this in context, so the exome, meaning the nucleotides that encodes for proteins, is about 30 million base pairs. And these have the very same single base that's been altered. There's now about 65 of these families worldwide that have the very same genetic cause. And the second family I just want to quickly bring up is this DNA GC13. So this is a Mennonite family from Saskatoon, which we in 2014, we published this paper where we found a single mutation that is transmitted with disease in its family. And we found an additional 5 smaller families you can see in the bottom here with the very same genetic cause. All of these coming from, 4 of these coming from Canada, and 1 of them is from Tunisia. And so this monogenic forms, so these genetic forms of Parkinson's disease, especially the familial forms, are rare. And I think they correspond to about 5, maybe 10% of all the patients that have Parkinson's disease. It's only a small fraction. But as I kind of went through, you can use this information for these patients to predict the prognosis and the symptom onset of the disease. And you can also actually study that, having this in mind, that this will probably happen, and then study changes that happens to the patients before it happens. You can also use this for clinical trials. But what I find maybe even more exciting is that you can maybe infer this information to then all patients with Parkinson's disease. And this is a classic picture that Matt keeps using in his presentation, but I think it's a pretty good way of illustrating this. Because we're looking at the brain, and we have ideas, but we don't 100% know what's wrong, what's happening that actually results in Parkinson's disease. But finding more and more of these genetic causes, we can maybe pinpoint the most essential where we should look and what kind of functions we should focus on, what kind of pathways or what kind of biology is important. And what we have found so far, and this is illustrated by the 2 last genes that I showed you, the VPS35 and the DNA GC13, is that they seem to converge in this trafficking of proteins, meaning that how proteins go from one part in the cell to another part of the cell. And with that, I'm going to leave that to Jordan to explain from those 2 models I just presented.
Okay, everyone. So we're going to switch from genetics and start talking about, I guess, the biology and the functional readout that we try and investigate from changes in DNA. So on your left-hand side, we have a simplified cartoon of the end of a neuron, of the terminal of a neuron, and the basics of neurotransmission is really the recycling of membrane. So there's this continuous recycling of material that's taken up into the cell it is sorted into particular networks depending on what that material is made up of. And then you have these very small vesicles that are filled with neurotransmitter. And neurotransmitter is obviously how neurons will communicate at a very rapid basis. And then when there is a particular stimuli that is received, whether that is a visual stimuli, auditory, or just general actions throughout your day, those vesicles are basically given the go signal, and they fuse with the cell surface or the end of the neuron, and it releases neurotransmitter. And if we blow up just a single vesicle to the right here, this is the membrane that our neurons are encapsulated by. We find that there is many constituents that regulate the movement, the lifetime, and the overall positioning and function of these vesicles at the nanosecond scale. And what's really interesting is that we're starting to find that a lot of the genes in Parkinson's disease that we believe are causal and pathogenic are situated in this terminal. So there's this very condensed hub of membrane trafficking, and I'm going to talk to you about how we take the genetics and we use mouse models and particular experimental techniques to explore how these mutations interfere with neurons communicating with each other in their lifetime. As Emil said, we've previously published on VPS35, which is a constituent and a main subunit of the retromer complex. And if we take one of those single vesicles, which contains our neurotransmitter, and we further enhance just a particular subdomain of that vesicle, we can see that there is many proteins that are situated all together, and that we have VPS35, which is shown in purple here, is part of the retromer complex. Oops. And then DNA-JC13, which is the second mutation I'm going to talk to you about, is situated ever so closely, and they form this quite large complex. And the best way to think about what this complex does is similar to a crane or something that will move cargo at a shipping yard. If you need to get large items from one side of a shipping or depot to the other, the easiest way is to use mechanical force to pick up that item and place it into another area of the space. And that's really what retromer does. It takes material that is taken up into the cell, and it sorts it into this tubule-like conveyor belt, and it will position particular endocytosed material along the conveyor belt for additional proteins to pick it up and move it along. So it's really about a very dynamic system that relies a lot on the movement of proteins. And so when we use high-resolution microscopy, you can see that this is a single neuron, and we've introduced two proteins into this neuron, one of which is GFP, which you can see will basically fill the entire cell. We have the dendrite, we have these little spines, and within each of these little spines you can see VPS35 will quite nicely will quite nicely decorate along the dendritic compartments. And we really believe that having a fully functional retromer complex within each of these little synaptic compartments is fundamental to neuronal function. Okay. So what we do is we make particular mouse models that will harbor a single mutation in the gene that we are interested in. In this case, it's the DNA-JC13 gene, and we've mutated a single residue, and basically we go about studying how the expression of that protein will alter cellular functions. And so similar to the previous slide, when we introduce particular proteins into the cell and look at it through confocal microscopy, high resolution microscopy, we can see that in the wild type or the healthy neurons, we see that our protein is quite nicely sitting and decorates the entire axon, and these very tight sphere-like structures, which are the little compartments we described earlier. However, in our mutant mice, where they carry a single mutation, we find this quite interesting phenotype where instead of being this very discrete, tight sphere, we get these rather elongated structures that kind of remind you of like a worm. And we see that there is this statistical difference between the percentage of structures that we class as elongated as opposed to spherical in the mutant mice compared to the wild type mice. And what I alluded to earlier is that the tubule is really important for the sorting and the positioning of endocytosed material. And you can imagine that if you are unable to, I guess, appropriately handle these tubules, they don't function as one would expect, and that really impairs the sorting of endocytosed material. So if you take away half of the garbage trucks, you're going to start building up trash and things that you don't particularly want to hold on to quite quickly, and that could be a very toxic cascade for cells that are very sensitive to waste. And then we take, I guess, the mouse as a whole, and we place them into this, pardon me, this open box, and the mouse is free to roam, it's free to explore, it's free to groom. And what's quite interesting is that at 3 months of age, so the mice are quite young, their ability to walk around and to roam and the speed at which they travel is consistent between wild, healthy and mutant mice. As is the number of times they will stop on the spot. So as opposed as a, I guess, comparison to the purpose of freezing, which is often seen in patients. However, at 9 months of age, we begin to see these quite nice differences between the healthy and mutant mice. So the distance traveled begins to become a little bit less in the mutant mice, as is the average speed that those mice, I guess, roam. However, what's kind of interesting and quite nice in terms of recapitulating and modeling the disease, we see that the mutant mice begin to have far more episodes where they pardon me, the mutant mice will begin to have far more episodes where they just randomly freeze on the spot, and that is basically I guess a nice aspect of the mouse model that recapitulates particular aspects of the disease. So this is really interesting. We're still working on this. We're still trying to work out what this means, but it's very promising that the mouse model that we've generated and engineered is quite similar to the human disease, and that's important for us in understanding how the progression of the disease occurs. So now I'm going to switch to a complete study that we've published, which was in 2018. Excuse me. This is available online. Everyone can look at it and download it if they wish. And this was a characterization of the mouse model of the VPS35 D620N mutation, which is what Emil described in the Austrian patients. And there are several patients in Canada that contain this or harbor this mutation. So I'm going to talk to you about that study. So the first thing we wanted to really know in these mice was is the number of neurons that produce dopamine different when the mice have a mutation? And the simple answer is no. So when we use slices from the mice, and we look at mice that are healthy, they have a single copy of the mutation or 2 copies of the mutation, we find that the intensity of the terminals along with the number of cell bodies in these mice, and this is specifically in mice, or in it's just staining for the dopamine-producing enzyme tyrosine hydroxylase, which, just to remind you, in healthy individuals, you see this black pigmentation, and in PD, late-stage PD typically, you see this complete loss of pigmented neurons. This is what this translates to. So this staining in green that we use an antibody against is investigating this same population of neurons. And so this would indicate and tells us that at 3 months of age, these mice do not have typical signs of neurodegeneration. It may occur later in the lifetime of the mouse, but at 3 months of age, when the study was conducted, the mice do not have gross differences in the total number of dopamine-producing neurons. But in saying that, what we then went on to investigate was the amount of dopamine that is being released. So dopamine is obviously pardon me, one of the fundamental neurotransmitters in the brain, and is obviously very heavily implicated in a lot of the symptoms that come with Parkinson's disease. And so just basically, when we look at the wild-type mice in white, and this is measuring the total amount of dopamine that is released from the cells, which is depicted in this cartoon, we see that the mice have an average of about 0.1 micromolar being released. However, what was quite remarkable was that when we looked at our heterozygous, so a single copy of the mutation, or homozygous, 2 copies, we found that there was almost double the dopamine being released. And that's quite phenomenal, because it again speaks to the importance of, I guess, modeling the disease progression in these mice. And this is also just quantified here, showing that this is this almost stepwise increase in dopamine release. And we have work that is ongoing trying to understand whether this will begin to decline as the mice age, which typically happens in patients with Parkinson's. And then we were really intrigued by that result, because it spoke to the, as I said, the modeling of the mutation in the mouse. And we then went on to look at whether or not particular proteins that are important for the reuptake of dopamine and the release of dopamine were different. And so when we look at this particular protein that I've highlighted in green, it is the particular transporter that sits on that little vesicle that I explained earlier, and it pumps dopamine into the vesicle. It houses dopamine. And what we found in these mice, although they had completely normal numbers of dopamine-producing neurons, we saw this stepwise increase from heterozygous and homozygous carrying mice of this particular protein. And then conversely, we found that the transporter that is needed to take dopamine up from the cleft, so once it's released, you want to have a very efficient mechanism to get it back into the cell and repackage it. And that's how you allow neurotransmission to occur very quickly. We found that the levels of this transporter were significantly down in the heterozygous and homozygous mice when we compared that to the wild type, indicating that the mutation is quite specifically altering the dopaminergic neurons at the I guess the terminal, at the business end of things. However, the total number of neurons, dopamine-producing neurons, was normal in 3 months of mice, mice 3 months of age. And we can see that particular other proteins across our different genotypes are all normal. So if we saw a loss of all those proteins, it would indicate that there is something very wrong with the mouse in terms of maybe its development or in terms of the gross number of neurons. But since we see these particular changes in particular proteins that are within dopamine-producing neurons, whereas other ubiquitously expressed proteins are normal. It speaks to the specificity of the mutation in dopaminergic neurons. And then we're still characterizing this phenotype because it's really quite interesting to us. But what we have also found is that when we take tissue from mice, or we use immortalized lines, we can show that VPS35, which is a constituent of the retromer complex, it will physically bind to the dopamine transporter. So this is an interaction study that we can do. And we're currently doing this in the mutant mice to see if the binding between our retromer complex and our dopamine transporter is different, and whether that binding difference may explain the loss. So if there is an absence of binding because of the mutation, or diminished binding because of the mutation, it would explain that the retromer complex with the mutation is defective in appropriately recycling the transporter needed to reuptake dopamine. So that's really exciting that we've made that link, and we're still working on that, and that may be reserved for next time. And so we were really interested in those phenotypes, and that was all published. You can read about that online. We could always send out the link to anyone that's interested. We were really curious if we could use commercially available compounds to rescue this phenotype. So if we feed the mice this compound in their food, or we give them an injection, can we change the results based on the amount of drug they're receiving and their genotype? So just very basically, when we take our wild type mice, you can see in the gray, and this is again measuring the amount of dopamine that is released, we see that it's about 200 nanomolar, sorry. When we take our mutant mice, which is in peach, we again see they have far more dopamine being released. And then when we take our mutant mice in maroon, and we give them this particular drug for a period of 7 days, the amount of dopamine being released is near what we see in the wild type. So this is very early data. We're quite quickly working on this to ensure it's correct. But what we find is that when we have this particular mutation in VPS35, which I've described, and we give them drug treatment for 7 days, the mice are completely normal, they don't lose any weight, and their behavioral aspects are okay. We can see that between the no drug and the drug, there's almost about 50%, 80% reduction in the total amount of dopamine that's being released. And that's really, really, really interesting to us, because it speaks to the importance of another protein that Emil has already alluded to, LRRK2, in regulating these phenotypes. So we believe that these pathways are inherently linked, and we believe that if we really have the opportunity to push this further, there will be some very fantastic data that may be available for the public, and will really help patients with VPS35 D620 mutations. So I think I'm running out of time, and the reason this is so interesting is because over the last probably 2-3 years, there's been this increase, this immense increase in the interest of LRRK2 kinase inhibitors in Parkinson's disease, and this interest is only going to keep growing, we hope, and we feel that we're on the right track. And what's really neat about particular LRRK2 kinase inhibitors is that there's a lot of work ongoing where people from other labs have taken just a standard old blood sample from the arm, and they've treated those blood cells with LRRK2 kinase inhibitors. This is a different one from what we use. You can see here that 6 random donors, so 6 people from the chat today could have their blood taken, and they receive what's referred to as the placebo or the control or the drug, and you can see that it completely abolishes the activity of the protein. And this is really quite a nice step forward because it speaks to the ability to very quickly and in a non-invasive method really assay particular compounds that may be specific for particular people with a very unique disease. So that's really interesting, and that's really where we're leading. That's where a lot of other groups are going. And yeah, as simple as having your blood withdrawn and then testing those red blood cells with particular compounds is really a fascinating aspect of disease, and we hope it will continue. So that's all from us today. We're 2 minutes over time, but again, this is where we work. This is the team. We have a lot of great financial support. We know our talk was very heavy on the science, and if anyone would like to ask any questions or come and visit the lab, you're all welcome. Matt, who is our boss, is very accommodating, and as are we. So yeah, please ask away or email us, or come and visit the lab. There's a lot to see. Thank you.
Yeah, thank you.